สวัสดีค่ะชื่อรัญชนาใช่ไหมคะ
ใช่ค่ะ
อ๋อค่ะสวัสดีค่ะ Good afternoon. I am Dr. Dongruti Thanramlit. I'm teaching you basic bioinformatics in MDMG 500 today. So my full name, Dr. Dongruti Thanramlit. My nickname is Ajarn. I'm teaching at Institute of Molecular Bioscience. If you have questions regarding to the course, you can email me at dongruti.tan@mahidol.ac.th. The class materials on my website can be found at http://ajanri.wordpress.com. So let me introduce a little bit about the class materials. So we have the lesson plan, which the lesson plan including the basic learning objectives and the references to the class. You can also download the learning materials from this QR code. The exam will consist of 3 questions, but before the end of the class, could you please do the online quiz together if we have time? So the deadline for the online quiz is 6 o'clock today. I will introduce you a little bit about my website. My website is ajanri.wordpress.com. You can come to courses, and these are all the courses I open to teach this year. So basic bioinformatics is this one, MDMG 500, and you can see the slides here. So the slide will be exactly as I'm teaching today in the PowerPoint slides. You can also find it on the Google Classroom too. So I also have the assignment gallery, which I taught over the years. So if you come to the class, we should have a lesson to make physical model of the protein and make some CRISPR-Cas9 poster. So that would be in the next session. So this session we will start with basic bioinformatics. The references to this class is Atlas book on Introduction to Bioinformatics here, and an article by Lescom and Greenbaum, What is Bioinformatics? And another book called Structural Bioinformatics by Philip Bond and H. Wilczek. So this is what the book looks like. You don't need to go to those books because I have digest all of them and put in the class materials that in your handout. The objective of the class, at the end of this lecture, you should be able to describe principle and flow of bioinformatics data necessary for research, identify bioinformatics tools used for sequence and structure analysis, and describe the methods of using bioinformatics for research. So the content of the class, it will consist of the overview and the central dogma and bioinformatics information, the type of bioinformatics data, including of nucleic acid, protein structure, structural data, and databases, and structural bioinformatics data, and application to other bioinformatics tools. So in modern day, we should learn structural bioinformatics at the same time with the science that we are studying. Please look at this video of what the future education should look like. We should see everything in 2 pictures. these pretty pictures are building from the files, like animation files, but the PDB files will be the format that used for preparing the protein structures. So I will introduce you later in the class about the basic structure in the PDB format. The concept in bioinformatics gathering around the central dogma, which is the DNA transcribed to RNA and RNA translate into protein. So the central dogma organized around the cycle of passing genetic information when DNA transcribed and the triplet codon specify each of the 20 amino acids. After translation, protein sequence with long polypeptide chain are synthesized. So let's have a look at this video about the details of the genetic code.
So DNA, since it's double-stranded, we don't use both of those strands in order to do transcription. One of the strands is called the template strand. And to figure out what the template strand is, mRNA is always made starting at the 5' going towards the 3', because that is how the enzyme RNA polymerase works. It always goes 5 to 3. So because our mRNA has to be 5 to 3, the DNA that we use as the template has to be anti-parallel or opposite. So we will use the DNA strand that goes 3 to 5. In this example, it's going to be the top DNA strand. So we're going to create our mRNA using complementary base pairing. And it's going to be running from 5' to 3'. So you'll notice in our DNA molecule at the very top we have G that will pair up against C on our mRNA. A in our DNA will pair up across from U. T is across from A, and then the C's will be across from the G's in our mRNA. So the really important part, or the biggest mistake I see in this transcription sequence that happens first, taking our DNA, creating an mRNA, is that people forget to use the uracil. So remember, DNA has thymine along with the adenine, guanosine, and cytosine. RNA has uracil along with the adenine, guanine, and cytosine. So this is probably the biggest mistake that I see during transcription. Other than that, it's pretty easy. You take your DNA going from 3 to 5 to create your mRNA, which will be going from 5 to 3. So they're, again, anti-parallel to each other. And then put uracil in when you're doing that complementary base pair. Then once we find the start codon, and there can be additional nucleotides in front of the start codon, those we can just ignore, because we always have to start with the start codon. Now we can go through the translation process. The translation process is a little bit more complicated. There's going to be another molecule called a tRNA that comes into play before we can start to put our amino acids together to make our protein. Alright, so what we have to do first is we highlighted our codon on our mRNA. We then need a tRNA molecule, and the T stands for transfer. So transfer RNA molecule, it's just meant as an intermediate to help read the mRNA and to bring in the correct amino acid to make our polypeptide. So on our tRNA molecules, which I have represented with the little 2 blue triangles now at the bottom, on that tRNA there is a 3-sequence nucleotide sequence called the anticodon. So the anticodon is going to be complementary to our codon. So in this case, if our codon is AUG, the start codon, our anticodon on the tRNA is going to be UAC. So remember, we're making RNA, so we're still going to have uracil, and then A's and U's pair up, G's and C's will pair up with each other. Okay, so now we have our first tRNA molecule with the anticodon, In order to find out which amino acid the tRNA is actually bringing in, or what it's being put first, is we go back to our codon up on our mRNA molecule, the AUG, highlighted in the orange circle, and we're going to be using a genetic chart in order to decode our codons. Okay, well first, this is the start of our polypeptide. So we go to our next codon, which is UCC. So we bring in our next tRNA molecule that has the anticodon AGG, because they're complementary to each other. And then to figure out which amino acid is coming in next into the second position, we go back up to our codon, which is UCC, go to the chart. Our first letter, if we look on the left, is U.
So the reason why I show this again is because the focus of bioinformatics 2 is mainly gathered around translating the DNA to RNA sequence to amino acid sequence. So we will have several tools for converting between each of the two molecules, like DNA to RNA, RNA to protein, protein to some other things. So in bioinformatics tools that I will teach today, such as in BLAST, you can see how to search the structure if you have protein, how to search for mRNA, something like that. So this central dogma was taught so many times, but these letters has an importance in bioinformatics, and you don't need to memorize all this because all the tools will have done it correctly for you. But you need to understand first that you need to have the sequence, and then you can go to format in other molecules. So the protein, once the RNA translates into protein, it will be a long string of polypeptide chain we call the primary structure. And then the primary structure has repeating torsion or repeating angles, which will be secondary structures. And if the secondary structure are combined to many units, such as beta, alpha, beta, or alpha, alpha, it will be called a motif. Once the motif become a recognizable fold that we can see in daily life, such as horseshoes, solenoid, sandwich, boxes, it will be called a domain. And the functional form, the most functional form of the protein will be tertiary or quaternary structure. So the tertiary structure already in working function, but in the case that it requires more cooperativity between each units of tertiary structures, it will be called quaternary structures, which make the protein more robust in order to function. So the basic building block of protein should be emphasized a lot in Ajahn Chanan's class already. Basically, the protein chiral molecule have the L amino acid chirality, and the torsions and everything should be mentioned in other class already, so I should go quickly. So in bioinformatics, the most important elements in the periodic table are along the second row. So if you plot the periodic table into a molecular visualization platform, you can see atoms with various colors and various radii. The most important one is in the second row for bioinformatics. So the colors that you need to memorize is carbon in white, nitrogen in blue, oxygen in red, and sulfur in yellow. So this is called the Corey-Pauling-Cotton color space. So the amino acid side chains, which taught early on as a drawing, but this one I will emphasize that when you actually look into the X-ray crystal structures, they have shapes. So glycine will be the smallest one. Alanine, valine, and leucine, you can see they have no colors. So these are aliphatic small amino acids. And if it has one red tips, that means it has OH. So serine, threonine have one red tips. You can see that they are crucial in kinase, such as in serine threonine kinase, because it allows for phosphorylation at the hydroxyl group. So once you can memorize the color code, you don't need to know the name of the protein, but you can infer the function by feeling the structures. Cysteine has sulfur, so it's yellow. Aspartate and glutamate have two red tips, so it has double bond OOH. In double bond OOH, it means it is partial acidic, so it has partial negative charge, and it's allowed for salvage with the positive charge molecules. And if it has one blue, one red, so it becomes polar neutral, polar uncharged, asparagine and glutamine. So aspartate and asparagine has the same shape, but it has different element colors, so it has different function. Glutamate and glutamine has the same shape, but glutamate is partial negative, but if it has one blue neutralized with one red, it becomes polar uncharged. So from the methionine, you can see it has a yellow color, so it has been used in solving structures such as in methionine. And lysine and arginine have only blue colors, histidine too. So these are positive charged amino acids. So it pair with the negative charge, pair with the red tips. And if it has 6-membered ring, it is aromatic amino acids. So phenylalanine, tyrosine, tryptophan are used by chymotrypsin to recognize large hydrophobic amino acid in the pocket, for example. And proline introduces king in the helix because it's rolled back to the backbone. So if you can see the colors and can see the shape, you may not need to memorize what the name is called, but you will know what the functions are. Like lysine and arginine used a lot in epigenetics is because it have partial positive charge. You can see from color blue. So if I have exams for you to tick boxes, which amino acids have certain function, you can infer the function by the shape and colors that you see. So the protein size are varies. Sometimes it's as small as trypsin inhibitor, which is 8 kilodalton, have 36 angstrom across, or it can go as large as ribosomal subunits, or even virus have megadaltons of molecular weight. So the molecular machinery, this is the chart from the protein data bank, which show various shape of protein in the protein data bank. You can see virus in spherical shape. This is GOER, so it has a barrel-like structure. When the molecule comes in, it has the lid to close it, and it measuring the protein to have a correct fold. Some protein have the top function like that. Some protein located on the membrane called transmembrane protein, like this one, ATP synthase, which I will show you in another videos. And the last one, like structural protein in microtubules. So proteins comes in various size and various shapes based on what function it will perform. The types of bioinformatics data. Normally, if we talk in terms of text processing, the DNA sequence are 4 base letters with length around 1,000 letters per genes, and protein sequence are strings of 20 different amino acid one letter code with about 300 amino acids or more. And if we talk about the structural data, it's normally in the PDB format, consists of atoms with XYZ Cartesian coordinates with several parameters in the back. So DNA sequence data look like this. This is we call the FASTA format. You have the greater sign and have IDs. So bioinformatics data are normally encoded by IDs, and these IDs, when you search in the databases, it can link to one another based on the function. So these are other details of this protein on the chromosome 7, and after the header in the FASTA format, it will be followed by the 4-letter code in DNA sequence. And the place that you can look at the DNA in the location of the genome would be called a genome browser, such as this one, Genome Data Viewer from NCBI, or you can go to Ensemble. So the usefulness of this web server is that you can search for similar genes for annotating introns and exons and link specific genes to the disease. The protein sequence data look like this. So this one is again the FASTA format. We have the greater sign here, and then followed by some codes. This one is the UniProt code. I have the UniProt name, cytochrome C human, and then it follows by the amino acid sequence letters. So homologous protein sequence are used for making multiple sequence alignment to find the functional size and protein modeling. This is what we call the multiple alignment. The aim is to align similar amino acids. You can see if it has the red colors, it have the charge, like this one, K lysine have red colors. And if it's hydrophobic, it has the cyan or the color of the sky. So basically, you have a scrambled piece of FASTA format, right? And once you put in the program such as cluster X, the cluster will basically insert the gap and then align all the amino acid letters with the same function in the same blocks. So I will show you later on. It's just one button click in how to do the multiple alignment, but I will introduce the various kind of data first. So for the structural bioinformatics data, the pretty pictures that I showed you earlier in the video built from the structure in the PDB file format. Basically, the PDB or protein data bank file consists of the header, and the header will say what the proteins are and has the title of the paper, like this one, protein kinase A in complex with a drug called stereosporin, and it will say whether the protein was engineered, what EC or enzyme commission number they are, and what organisms of the protein that it comes from, and the expression systems that they use, like this one from Bostoras or from cow, and they express it in Escherichia coli. So header is an important thing. The juicy bit, the most important thing in the PDB file format is this chunk. We will have the atom records in the first column, and then have the serial number running, and then saying what atom type there are. So this one, nitrogen, C alpha or alpha carbon, carbon of the main chain, oxygen of the main chain, C beta, which is the C carbon atom next to the alpha carbon, and C gamma next to the beta carbon, for example. And this column called residue name. So this one, it groups the atoms together into these residues called glutamate. Glutamate has chain E, and this is the residues number. And then it followed by X, Y, Z coordinates and other terms, which I will be covered when we talk about the X-ray structure if we have a chance. But for now, let's look at the glutamate. So this one is what the glutamate structure when it draw into chemical drawing structure look like. So it will have alpha carbon and going along until C double bond OO. So this is O epsilon 1 or epsilon 2 like this. And then the next one is called histidine in the same chain. So once you got that kind of information, you can plot the line format of the structure, or if you make the atom bigger, you have the ball and stick model. So ball and stick model, they draw the bond as stick, and then they draw the atom as the big ball. And if the radii of the ball and stick are equal, it will be called the stick model. So the stick models use a lot in X-ray structure drawing. So another one, which is called CPK space fill. So they use CPK color that I showed you earlier on, but the space fill basically have the radii of the sphere equals to the van der Waals radii. So the CPK space fill or the surface model truly represent the active site boundary in the cleft of the protein when we design the drugs. So this one is easy to draw by hand on the paper. Stick model used a lot for X-ray crystallography surveying. Ball and stick used for like chemical synthetic purpose or other things, and CPK space fill used when you want to design something that require the correct radii of the atoms, like in drug design. So let me introduce you a little bit more. After we have the line representation, and then we have the alpha tracing of the line representation based on the main chain of the protein. If we omit all the side chain and only retain the main chain side, you can see the overall organization of the protein structure like here. See alpha traces, basically they draw all the tracing of the alpha carbon. And if you color it by different chain, you can see how many chains there are for one structure. So this protein has 6 chains. You can see that if we color by chain, we can see 6 color really clearly. And another popular style is ribbon or cartoon. Basically, they use the ribbon to represent the alpha helix and beta sheets. And then there is the schematic and van der Waals. I will show you later. So this one, we see alpha trace. We omit all the side chain. We have alpha trace. The ribbon, basically, the trace along the main chain. And if we draw line more repeatedly thicker and thicker, you will have the spiral shape of the main chain, and the molecular visualization tools will add light and shadows until you see the pretty pictures. And if it become turn like this, they will color it like green, different from the strand beta sheet, which color by cyan. So this is cartoon representation. The schematic representation is used when you want to see the direction of the main chain, where it's coming from. So this is the rocket shape. From N terminus to C terminus, you can see the rocket. This one represents the alpha helix, but the draw is as a rocket altogether. And this one in arrows. So this one go from N terminus to C terminus. So this one you can tracing around the N terminus to C terminus from alpha helix and beta sheets. So you know the main chain direction. You can trace where it starts and where it ends if you use the color by chain representation to see the gradient from red to blue. So this one is the final style called van der Waals surface. It's basically draw all the atoms with van der Waals radii. And if you color it by charge or pKa, you can see the color of negative charge amino acid in red, positive charge amino acids in blue. And basically, when you want to design molecules to catch with these kind of proteins, you will design something opposite. Like if you want to catch with red, you design something with the blue. So when we talk about the virus, like coronavirus, adenovirus, this is what the structural biologists feel. So basically, the structure of virus comprises of proteins and genetic materials, RNA inside. I will show you an interesting video from Janet Iwasa showing the detail from PDB files of the virus. So this is the virus, has the capsid RNA inside. This is the size of the HIV virus in comparison with the T cells. So you can see that virus is very small. Once it wants to enter the T cells, it will rolling along the surface of the T cells. So upon rolling, once it finds the receptor, it will stop. And then when it tries to infect the T cells, it will have recruit cellular molecules in order to insert the genetic materials inside the T cells. So it punched the hole with the red molecules here. So you can see alpha helix, which it used to punch the holes. And then it seals the two membranes together. And then the genetic materials of the HIV is pushed inside the T cells. So this is what I trying to introduce you about when you want to design drugs or want to find drug target. Basically, you look for the receptor first. What will be the protein target that you want to attack? So in this case, the design peptidic ligand in green. So it mimics the peptide, but it doesn't perform the function as in blue shadow. So it just wrap around and stay there. So by wrapping around, it stops the virus from entering the cells because it just stays in that place, but it doesn't allow for the sealing, the fusing of the two membranes. In a nutshell, so the phenotypes is the result of environment influence combined with the genotypes. The genotypes with mutations then pass on to the subsequent generations, with beneficial mutations being retained through evolution. Some DNA mutations are silenced, while some others can cause aberrant function. So we assume that a pair of similar genes give rise to similar protein function if the genes are diverse from common ancestor. So let me explain it more clearly. Once you have multiple alignment, these are unimportant things. So the loops is basically specific to corn or sunflower, but the important function that return to millions of years of evolution is in the conservation of the blocks. So basically, when we have certain sequence, we want to find these blocks in comparison to all others that exist through evolution, and these blocks to do further experiments, such as proving the function of the domain blocks. So it comes to the concept about homologous protein structures. So if the homologous proteins are diverged from one another after speciation into different organisms, we call orthologs. What orthologs mean is that if you have protease for virus, protease for other organisms, or if you have the enzyme like lysozyme from birds, lysozymes from cow, lysozyme from monkeys. So if they are all lysozymes, we call orthologs. So it means it's the same enzyme that exists in many species. So these ones, once you got the sequence from one organism, you can blast or can search to what the sequence looks similar to in other organisms. The other concept is what we call paralogs. So paralogs are related as a result of gene duplication. So it's become the species already, and then once in that species, it diverges from one another again, like enzyme in various isoforms, but in the same organisms. So when we find the orthologs or paralogs, we basically find in the sense that amino acid sequence similarity is very high. So once the sequence similarity is high or sequence identity is high, we infer that it adopts the same protein fold with the known genes. So homologous protein can be either orthologs or paralogs. Orthologs exist before speciation. Paralogs exist as a result of gene duplication after speciation. Okay, ก่อนนะ So next part, I will teach about bioinformatics tools and database widely used currently. So the DNA sequence can be obtained by this animation process. Please have a look a little bit.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
So the tools that we use to search the sequence is called BLAST. BLAST stands for Basic Local Alignment Search Tools. So once we have the amino acid sequence or the nucleotide sequence, you can use nucleotide BLAST to search the DNA or RNA with the DNA or RNA, so nucleotides to nucleotides. If you have the protein sequence and you want to know what nucleotide sequence look like, you can use T-BLAST-N. And if you have protein and you want to search other proteins, we use protein blast. So it's not only converting in the sense that once you submit the protein sequence, you can search the whole database. Whatever everyone in the world have done and deposit, you can see what the protein looks like in the form of nucleotide sequence. Let me show you how. So basically, if we have FASTA format, have the text look like this, just that text file, have greater signs, and then some IDs in one line, and then amino acid sequence. So you see this amino acid sequence, just short ones. So you have a text file with 3 lines in Notepad, and we save it as FASTA. And when we blast, we do something like this. We go to blast at NCBI, and then input the 3 lines in, or you can use it as the text file and say as file name, human cytochrome C.FASTA with these 3 lines, and we blast. After blasting, you can see records of this one cytochrome C as input in amino acids, and you can have series of mRNAs that corresponds to this chunk of amino acids. So once you search and then you click in any items, you have this origin region of mRNA. So this is how you use bioinformatics to discover what other nucleotides look like this queried protein segments. So this is how to blast. It's just easy ones, but when you search in real time, it takes a bit long. Basically, it will have scores. Like here, you can see 1e to the power of minus 70, 100% match, something like this. So you basically look for e value very, very low, like 10 to the power of minus 70, 80s, and 100% identity. So it means it's exact match of what you have searched with your FASTA files. So DNA have 100% match just means it every letter will translate directly without mutations. The next resource I will teach is called UniProt KV. Okay, when you go to the UniProt, you can input cytochrome C human, and then it will show you the function, electron carrier protein, and then it shows you the binding site location, which amino acids there are, and the gene ontology, which is the specific terms used for describing the molecule's function. This can also show you the subcellular function location, such as this one, say it is in mitochondria. And UniProt offer the list of chemical compounds that are found inside the protein pockets. And it shows you also the structure of the protein. So this one, you can see the structure of the protein here. But it's not proper tools to do the structural analysis, right? It just shows a brief introduction, and this is FASTA files. So it also shows mRNA translation sequence. So you click on any IDs, it will direct you to that database with the association of the molecular function. So basically, bioinformatics consists of various IDs like this. When you go to the website, if you see the colors, it means this one already matched that it's associated with your queried sequence. So in this case, we queried for cytochrome C human or P99999. And then any other IDs here will be correlate, like relating to, like this one superfamily that relates to your sequence. So you can also use UniProt to blast. So you put cytochrome C human like this, and you can blast inside the UniProt too. Various tools allows you to blast for proteins inside the database. So this one, you don't need to put the sequence, you just put the IDs, and it shows you, let's say this one, cytochrome C of mouse and rat. Ratus novaticus is rat, Mus musculus is mouse. So it shows e-value. When it shows the match, if it has high matching percent identity, it means e-value is very low too. So it's 2 to the power of minus 67. So the primary database for structure is called RCSB Protein Data Bank. In Protein Data Bank, it will consist of 4-letter code that you can search to the protein structures. PDBE is a European version of RCSB Protein Data Bank, so it stands for Protein Data Bank in Europe. It uses the same IDs but offer different presentation. You can also download the PDB files from this PDBE or RCSB Protein Data Bank. The file will be exactly maintained to be the same. So this one, PDBE and RCSB, are primary data source. The primary data source means that the X-ray crystallographer solve it and then deposit it directly by the maker of the structure. So it's without process, but it finds other information that the raw format, the main structure comes from the experimentalist who has discovered them. So this is the example of primary data source for protein structures. So once you come here, you can put the protein name, or you put UniProt code, BRCA1 human, or you can put 4-letter code. 4-letter code, basically the PDB file format will come with the 4-letter ID. These 4 letters are unique identifier of that protein structures. So in this case, one STC can be used widely across all the protein data bank resources to basically means cyclic AMP dependent protein kinase in complex with styrosporine. And you can download the structure in PDB file format from this button. PDB-SUM is another flavor of protein data bank in Europe. It allows you to put the PDB code or put the amino acid sequence here in the box, and then it offers you list of the structure that match with that amino acid sequence. So in PDB-SUM, it will show you the amino acid sequence and also have pretty pictures to describe which one is alpha helix, which one is beta sheets, and then it shows you the ligand binding site. So that's the example of primary data source for protein structures. The next type is called derived database or secondary database. So example that I show here is called the database called CATH. CATH stands for Class Architecture Topology Homologous Superfamily, CATH. So you can see these kind of acronyms across various bioinformatics data sources. They will have full name, and then they use the first letter to come together to form a pronounceable name. In Cas, it processes the PDB and group them based on the architecture. So once you have the protein structures, if it is mainly alpha, it will have letter 1. If it is mainly beta, beta sheets, alpha helix, beta sheets, if mainly beta sheets is called number 2. If it has mixed structure between alpha helix and beta sheets, it's called number 3. And if it leading by letter 4, it means it doesn't have much of secondary structure. So once you get into Cas, you can see what I called the domain. So architecture or domain basically means it relates to what you recognize in daily life. So if the protein are mainly beta, have beta sheets a lot, and if it looks like a box, it will call 3.70. And if it look like sandwich, they will ask how many layers of sandwich. So if it has ขนมปังปูอัดขนมปังแล้วก็ทูน่าแล้วก็ขนมปัง that's 3 layers. So if it's only 2 layers, it has only cycle, something like that. So yes, 2-layer sandwich, 3-layer sandwich. See? So it asks first how many beta, how many, if it has mainly alpha, if it has mainly beta, and then what it looks like. So it's in the second letter, if it's 3-layer sandwich, 3.40. And then it comes to, does it have leucine repeat or something that called topology? And then after topology, it will come to the homologous superfamily or homologs of the proteins that's similar to others. So it has class, architecture, topology, homologous superfamily. So this protein has certain PDB code 1P9A. So this is the PDB code, and then it will have associating Cas ID. Like this one, and when you go across various databases, it will find 3.80, 0.10, it means this one is mainly alpha, mainly beta, and it looks like a horseshoes, have leucine rich repeat, and in homologous superfamily called ribonuclease. So far, is there any question so far? You can ask. Speak up. No? Okay, I'll go on. This one is tough. So the concept called ontology. Ontology makes you hops around various databases by it acts as a dictionary of terms that present into numbers. So ontology basically means controlled vocabulary. It's limited, explicit, and carefully defined sets of terms, important for distributing queries among several databases. So by using ontology ID, it allows you to batch query proteins with specification and implies biological meaning of protein across various databases. So what it is? So for example, this one is called ontology. We have GO, gene ontology, and then 0004672. When you have the IDs like this one called ontology, it shows you that this one is specific for the vocabulary called protein with kinase activity. So the reason why this one becomes useful is because ontology can have relationships among them. So ontology can mean protein kinase activity is a kind of phosphotransferase protein with this gene ontology ID and have transferase activity and then go up up with is a relationship up to the top of molecular function. So this is function in catalytic activity. So it reads from bottom to top. The bottom one is the most specific one, and the top one is the broadly categorized terms. So gene ontology have three main domains. It have molecular function, biological process, and cellular components or subcellular locations. Why it's important is that when you want to search for protein with kinase activity and you want the whole kinase activity in the world, if you Google it, you will find PDF, PowerPoint, DocX, whatever, and you can find all kinds of junk. But if you go by IDs and you go to the correct database, such as you go to RCSB protein data bank and put GO004672, what you have is exactly all protein kinase with kinase 4 with kinase activity. So it's become handy because it have relationships among terms. So it comes into relationship like this. From is a relationship from the bottom go up to the top. The top have three main categories: molecular function, biological process, and subcellular location. So if you want to search every protein in mitochondria, you go for gene ontology of mitochondria, and then you get every protein under that with the correct term. So you understand the basis of hopping around various databases, right? So it linked by having IDs, and IDs have relationships among them. Gene ontology is one of the terms that can group protein based on the function, on the process, or on the subcellular location. So after you get the IDs, let's come to the more complex visualization of pathways. Have a database called CAPE. CAPE is the real-time curation biological pathways database. So if you know the protein called P38, it will allow you to highlight the colors of that amino acid, that protein inside the pathways. So if you know the PDB code, you can do PDB code to CAT. Or if you know the EC number, enzyme commission number of the protein, you can do enzyme to CAT. And once you convert to CAT ID, so it has series of CAT ID numbers, you can highlight the nodes inside the pathways. And CAT have every pathways in the world into this kind of pretty pictures. So you can focus on certain signaling pathways like Krebs cycle, NADK signaling pathways, and then you can highlight which protein with which PDB code. There's all linking between PDB IDs with KEX IDs. So this is another one in KEX pathways. So we can see the oxidative phosphorylation pathways with various proteins. But that's the old style. So it's easy for web interpretation of various function. It just gives you overall impression as fast as possible. But when you learn oxidative phosphorylation in the future, it will be more elaborate into videos. So this one is my database. I create my own big data database called MONORA. So MONORA stands for Mapping Analogous Nuclei onto Residues and Affinities. It basically links the database from CAT, so system biology in CAT, with the drug discovery database, as in the Campbell database, and linked to UniProt PDBE open targets, and it linked to genetics. So this database link various databases together, and it used for drug design. But I will not ask you about my database in your exam because I think there's other more important question to ask. But I will demo a little bit how it works. So this is vanilla. So you can search for drug name tyrofluoroperazine. So this one is the drug to treat schizophrenia. So once you search the drugs that used to treat schizophrenia, you can see that it linked to this one UniProt ID and this one CAKE ID. So in CAKE, you can search to the function. It said this drug, trifluoroperazine, involved with calcium signaling pathways and involved with cyclic AMP signaling pathways and cardiac muscle contraction. So by having this kind of database, you can know that the schizophrenic drugs to treat madness also cause heart problems in cardiac muscle contraction. So this is just by linking database. If you actually treat the patients with the drugs, it will take years until the patients become infected at their hearts, and then we can learn, okay, this drug has side effects on the heart. But with this database, it links the database together, so you know right away that it involved with cardiac muscle arrest. So it also shows you the structure of trifluoroperazine inside the protein and dissect it into molecular interaction, like how many hydrogen bonds each atoms made inside the pocket of the protein. And it also linked to the protein and RNA baseline expression. It says, okay, this one goes to the heart and to the muscles. So that's basically what my database does. So it links among various databases together, and it also has in-depth function, which I will cover later on in drug design lectures. So information that can be derived from protein structure. If you have a protein structure, you can know the fold, and the fold such as sandwich, three-layer sandwich, relate to evolutionary relationship, how it relates to other species. And you can know crystal packing, which affects the interaction interface. You can know the location of mutation, SNPs, and conserve residues from knowing the protein structures. You can also know the protein ligand functional size, where the protein and ligand interact and at which amino acid locations. You can learn about the catalytic function motifs inside the pockets. You can know the electrostatic properties of amino acids inside the clef in order to design drugs. You can know antigenic size and surface patches. So basically, most of the drug discovery project geared towards knowing the target protein and their structure in order to design the correct ligand to do the proper function inside the pocket of those proteins. Okay, so the case study I use a lot in this class is protein called cytochrome C. Cytochrome C is an electron shuttle in oxidative phosphorylation. So basically, it located in the inner mitochondrial membrane. It shuttles the electron from cytochrome EC1 complex through cytochrome C oxidase. So if you study it in textbook, it looks like this, but we are computational biology person, and the study of cytochrome C should look like this.
The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
Okay, so the structure of cytochrome C look like this, and we have seen in the video. So we will study further closer to the architecture of the cytochrome C, which acts as an electron shuttle. So the size consists of 104 residues, and is mainly alpha helices and reverse turns. It's called orthogonal bundle, means it looks like a square basket. Inside, in the middle, it has the heme molecule. So this one is structural alignment. Structural alignment means you take the structure in 3 dimensions and superimpose it one to another. So this one, the pink one is yeast, the green one is rice, the blue one is tuna, and the yellow one is horse. So you superpose all of them, and it has the architecture that have high sequence identity, so it's almost superimposable among all. So this one is cytochrome C. And when you do multiple alignment, before doing a multiple alignment, when you get the FASTA files, the FASTA files look like this. So it's all scrambled. But after it's all scattered like this, you can click on alignment and just click the basic function, whatever the default values are, and then you get the alignment like this. So you can see this one is cytochrome C from human in the first line, guinea pigs, rabbits, and then if it doesn't look the same at all, it's sunflower or corn. So they insert the gap in order to make the amino acids that are more similar onto the same blocks. And the gaps are inserted to avoid the loops in other non-related structures. So the cytochrome C is very important in evolution because it's found to be developed early in life. So in evolution tree of life, cytochrome C happens in the very, very early, millions of years before here. So it's truly homologous, and variation is compatible with the non-phylogenetic relation. So if you want to know how the evolution among human, monkey are far apart from sunflower, you can use the cytochrome C sequence, which can relate to the evolutionary time. After you get the alignment, you can have this button called trees here. So if you click on tree, you can build the relationship with the alignment, and then you can see the phylogenetic trees of cytochrome C. So in this phylogenetic tree, the closer one that pair together means they are more closely related to one another. Analysis of phylogenetic trees has tricks. If you want to group the organism into various groups, basically you plot the phylogenetic tree like this in unrooted trees, and then you cut the trees. After you cut the trees, you can see that you can group animal or human or something. This one is fish, alligator, and starfish, bullfrogs. So by cutting the trees, you can group this one into evolution of marine life. This one is evolution of birds. This one is evolution of human, monkeys, and then there's plants here. So it's far apart, but it's grouped here. So that's how you roughly analyze the phylogenetic trees. And cytochrome C can be used to study the interaction sites. So mainly the top part of the cytochrome C is hydrophobic patch, and the bottom one is called cytochrome BC1 complex. In cytochrome BC1 complex, if you color it by charge, you can see a lot of red colors, which is negatively charged. And you can see that on the surface, the cytochrome C have positively charged in where it catch with the negatively charged red patch. So basically, that's how it hops from one another. So it attracted by different charge. If you want to build the model, for example, if you know a lot of cytochrome C structure and there is new organisms that you never find before in the trace of life, and you want to know how it relates to the evolutionary trees, such as if you have platypus, and you want to know whether it's like a mammal, whether it has sequence similarity to ducks, or whether it's a bird, you can use cytochrome C sequence of Tumpaktot, and then you can do evolutionary analysis by plotting the trees, and you can also infer the functions by doing homology modeling. Homology modeling is like you use all the structures which have high sequence identity and superposed together, and then you identify the homologs and retain the part of the three-dimensional structures that you want to use as the model. And after you design aligning the target sequence on the known structures, you can infer the main chain from them, and then build atomic coordinates of the core, the loops, and the side chains up. So you can build up the whole model by knowing that if it have high sequence identity to other organisms, and you build a new one, and it should have the same flow if it have high enough sequence identity. So that's how to make homology model. So this is called structural alignment to make homology model. So you borrow side chains and then you get a complete structures. But that's just an imaginary ones. The example I used to describe how to make homology model is like if you have the part that look like bird, and the part that look like a horse, and the part would look like a rhinoceros, rhino. So you build rhino horn and wings, and then horse body. You have a unicorn, but then unicorn may be existing in the world or may never exist in the world, you don't know. But basically you have roughly the idea of what it looks like. The next example would be protein kinase. So protein kinase used a lot in drug design because it has a friendly pocket, so the pocket a lot of molecules can get in and bind there. But it has double-edged swords because once it binds to too many things, it causes a lot of side effects too. But a lot of cancer drugs are built, are created to attack protein kinase. So the way to analyze the pocket, you may be able to find the conserved atoms inside the pockets and know which part should be conserved, which part should be designed in order to fit the drug better. So this is the kinase map that I create. So once you know the concept part, you know the part that should be kept constant. So this part of the drug should be kept, this blue tips should be kept because it have conserved atoms and residue to interact with it. And then you can learn which part should be expanded or which part should be contracted based on machine learning. So by having lots of protein structures, you can know which dimension inside the pockets should be expanded or should be contracted in order to bind the drugs better. So the details would be emphasized a lot in drug discovery class, but for now, this class should be at this slide. So in conclusion, information from DNA, RNA, and proteins are converted to computable data format for bioinformatics analysis. Data in bioinformatics database has unique identifier or IDs. And mapping these IDs allow data integration from various databases. In the big data area for biological science, a lot of efforts go into generating data, but much smaller amounts go to analyzing it. So with bioinformatics applications, you should know the insights into biological information that will enable the design and validation of experiments for bioscience. So we end the slides here.